Okay, let's start with the electron transport chain just for review. Make sure we're all on the same page, and then we'll get a little more in detail. So, just as a reminder, the electron transport chain is where we have electrons that we collected on NADH and FADH2 coming from glycolysis and the citric acid or Krebs cycle. They're going to transfer electrons to protein carrier complexes in the electron transport chain. And those electrons will move from carrier to carrier in the chain, releasing a little bit of energy each time until they end up attached to oxygen at the end, along with some hydrogen ions, which, when you put all those together, you end up with water, and that's why you breathe out water. And all of that's going to happen on the cristae. For ATP synthase action, as those electrons move down the electron transport chain, as they're passed from one protein to another, that energy that's given off is going to be used to pump hydrogen ions across the membrane. And when we do that, we create a really high concentration of hydrogen ions in the intramembrane space, that area between the outer and the inner membrane, which makes them want to diffuse back into the matrix. But the only way across the plasma membrane, since it's made up of phospholipids, which are nonpolar, charged ions can't easily pass through. So instead, they have to go through a transport protein or some kind of channel protein, and that's what ATP synthase is. It's a channel protein for hydrogen ions that happens to also have a molecular turbine attached to it, so that as the hydrogen ions flow through it, they spin the turbine, and the spinning of that turbine allows us to attach an inorganic phosphate to an ADP, which makes ATP. And we get somewhere between 32 and 38. It really depends on what book you read and the efficiency of the process. So that looks like this. So here's NADH coming in and dropping off electrons and hydrogen ions. So the electrons are going to go into the electron transport chain. The hydrogen ions will be here in the matrix. The electrons will fall down the electron transport chain from one protein to the next, and each time a little energy is released, which allows us to pump those hydrogen ions into the intramembrane space over here, creating a high concentration, which makes them want to flow back through. In the meantime, the electrons are coming down this chain, and eventually they land with oxygen because it's highly electronegative. It attracts electrons and combines with some of the hydrogen ions that are here, forming water. So those hydrogen ions that flow through, they're going to spin this little rotor or turbine here, and that's going to allow us to phosphorylate ADP. ADP is floating around in here. Phosphates are floating around in here. The energy from the spinning will be used to attach it, and that will give us ATP. And now the cell has ATP available. You can watch the video of this in the topics page. I'm not going to show it here in this video. A video of a video seems a little odd. So you can go there and look for it. So pause if you want and go check it out and then come back. That brings us to anaerobic respiration. This is when there isn't oxygen available, because if there's no oxygen, then there's nothing to pull the electrons off of NADH collected in glycolysis. So kind of the whole process shuts down. So lactic acid fermentation and alcoholic fermentation are adaptations to not having oxygen available to draw down the electrons. So we'll talk about both of them, but first just take note of the fact that lactic acid fermentation really is just glycolysis, and that's really what alcoholic fermentation is as well. The difference is that after glycolysis, the part that we know, will have another reaction which will produce lactic acid, and you'll see why in a second. In the end, we're going to end up with 2 ATP that we net. Alcoholic fermentation is really only going to be used in yeast and some other microorganisms. And instead of producing lactic acid, it's going to produce alcohol and carbon dioxide as a final step. So here what you see, this is lactic acid fermentation. So there we are with glucose. We're going to use glycolysis to wind up with 2 pyruvates. Everything here is the same as what you've already learned. But now we'll have one additional chemical reaction to change pyruvate into lactate, because when we do that, we regenerate NAD. In other words, in order to take pyruvate and make it lactate, we need hydrogen ions and electrons, and we get them from NADH, which means we then produce NAD+, which means now glycolysis can happen again because we have NAD+ available to pick up the electrons. So that's why lactic acid fermentation needs that extra step here. If we just stopped right here, we wouldn't end up creating any more NAD+. We'd just have tons of NADH and it would shut down the whole process. And essentially, that's what happens in alcoholic fermentation too. You can see we go from glucose to pyruvate, but now we have 2 extra steps. We're going to change pyruvate into acetaldehyde, and then we'll change that into ethanol. And when we produce acetaldehyde from pyruvate, we give off carbon dioxide. So that's where the carbon dioxide comes from. And then that final transformation to ethanol is where we regenerate the NAD+. In other words, we need electrons and hydrogen ions to convert acetaldehyde into ethanol. So hence why it's called alcoholic fermentation, and it gives off CO2 and ethanol. Hopefully that helps explain some of the stuff in the book a little bit for you.